today my topic is Biobank for personalized medicine. This means bench and to bedside. The bench is the bench, the DNA extraction and some other experiment bench. This bedside is the hospital. So the technology to the hospital. So the biobank is important, the bridge between technology to the hospital. So before biobank establishment, this technology and this hospital so separate. So someone developed very, very important and interesting technology, but the hospital doctors, I don't know what do you mean. So there are many barriers between the bench to bedside. So we need communication between basic research and the hospital. So the communication of basic research and the clinician is important. So that's important role of biobank. So this is actually come down to the right. This is the Time Youth magazine at 2009 October 12. They said that the 10 ideas changing the world right now. One of these, you can just read it. One of these, Biobank. At that time, I don't know why the Biobank is important, the change in the world, but you can see how biobank changes the world. So today my topic includes these subtopics. The one is the introduction of Korea Biobank, and number two is our biobank strategy and the introduction of our hospital biobank. And the third part is the cancer genome research related to biobank, and finally conclusion. Before 2008, the biobank system is individualized, not organized. Every hospital and every clinician, every research have their own biobank for their own purpose. But if I need stomach cancer tissue, do you know stomach cancer? In here, cancer. So for the drug development or diagnosis, we need a stomach cancer tissue. So ask clinician to take tissue from the stomach and store it. After one project, completion of a project about the stomach cancer, we throw away to the basket. So no one can use it. But after building of a biobank, we store it in the bank and we can distribute to the other researchers who need tissue for their research. So that's why we established the organized, nationalized biobank. So in Korea, two main biobank systems. One is the Korea Biobank Network, supported by Department of 보건복지부, Department of one is the Korean National Research Center, is differently supported by department. the Korea National Resource Bank is the Ministry of Science and Technology. They also focused not only human tissue but also other microorganisms, animals, plants, and the human origin and the fusion material. So in the Human Origin Bank, there are Korea Cell Line Bank and the Liver Cancer Specimen Bank, Korea Prostate Bank. But this Korea National Resource Bank is not focused on the only human and human originated tissue or cell. So 2008 we established the Korea Biobank system. Korea Biobank network composed of central bank. As you know, in Korea, Korea National Bank and many regional bank. You know the Kookmin Bank and the Nonghyuk Bank, same. So we have a central biobank and 17 regional biobank. So there are different roles of this bank. Central biobank mostly collect specimen, population-based biospecimen. Maybe you know the cohort. Do you know cohort? No, cohort. Cohort means some big group of people, such as in the people around the Ansan area or people around the Gangnam area. So we collect sample regularly, and also we collect some clinical information such as hypertension and such as diabetes. So we collect specimen and the correlation between some DNA genotype and the occurrence of hypertension or occurrence diabetes, occurrence some other disease. We call it a cohort. So this central bank focused on the population-based biospecimen cohort, and the regional biobank located in the hospital. So we collect disease-based biospecimen, cancer or other cardiovascular disease, some other disease. So central biobank collect 300,000 people, and regional biobank 200,000 participants. We collect specimen. So this cohort and the national biobank and the regional biobank, this omics, NGS and omics, we call it bioinformatics. So the tool is bioinformatics, and the specimen is cohort and disease regional biobank. In the center regulate these two biobank. This is a population-based biobank, and this is a disease-based biobank. This is the central biobank in the Oson. Do you know Oson? Oson near to Daejeon. That's you can see. You can go drive 30 minutes from here. So very big biobank built in 2011. So very beautiful and big central biobank. And there are 17 regional biobank. You can see the Jeju. We have biobank in the Jeju. Why they are so distributed in the whole nation? Because that's why in the disease study we need multi-center institution. Even though the Seoul and this area is a highly populated disease, but we need this wide distributed biobank. So in the center in Seoul 5 and here 5 and here 511. So we evenly distributed in the nationwide regional biobank. Sorry for the delay of this data, but 2011 we already population-based 368,000 samples, and disease-based over 200,000 samples we collect. And this is a cohort established by the Central Biobank. This is Ansan cohort, baseline survey, and the second follow-up, first follow-up started 2006 and 2001. We collect biospecimen. Also, disease-based biospecimen composed of mostly cancer specimen. Do you know why we collect cancer specimen usually? Let's think. Can you accept when someone said that I want your tissue? No. So when you diagnose as a cancer, mostly do the surgery. But when the cancer surgery, not only cancer itself, but also adjacent tissue in the cancer, we dissect together. So that's why we easily collect cancer tissue. So when you do surgery, if you have cancer in the stomach, surgeons do not excise tumor only, but also your normal stomach also excite. So that's why we collect mostly cancer and adjacent normal tissue. And the other is digestive disease, and the other is allergy disease. The biospecimen, this distribution shows. And for the good biospecimen, we need quality control. For the quality control, we have various QC program for DNA, DNA purity, and RNA. We do the quality control program. So also we distribute biospecimen for the research. 578 project distribution from the Biobank. So mostly Central Bank, also the regional Biobank, we distribute many vials of tissue and the serum and the plasma for the study. The number of distributed sample from 2008 over almost 300,000 vials. So this is the Korean Biobank project. Is strategy like this, and the goal is this, and the vision is this. Actually, we are three stages of Korea Biobank. This stage is the second stage. Next year we move to third stage. This is the introduction of Biobank and the Korea Biobank system. And now I will talk about our Biobank. Actually, in my hospital, Pusan National University Hospital, we didn't have previously not good Biobank. So I was so stressed to make a new Biobank. So for this, I need a strategy of Biobank. So that based on the demand of customer. every bank needs demand of customer and the environment of biobank. What is demand? Demand is R&D use, research use demand, and environment biobank. I can explain environment. If some researcher ask me, I need liver tissue, but in our hospital we don't do the liver resection, so I cannot give them liver tissue. So I have to know what disease distribution our hospital and what demand of research. So I can tell you this Chinese character like this. GP is environment of biobank. GP demand of customer. GP environment of biobank. You can understand. So at first I want to know what's demand of customer by the PubMed research, because every people, every professor wants to publication of journal, and I searched the Korean journal with the trend of research, and the research composed of mostly neoplastic, and the organ is GI. GI means gastrointestinal, stomach and colon like this, and the breast and the lung. GYN is gynecology, uterus, something like, and the leukemia. This is neoplastic, and this is non-neoplastic. Immune disorder, cardiovascular, and the infection and the nervous system, something like that. This is the Korean journal, trend of research. And this is the international journal, submitted by the Korean also. So this is the Korean research trend in the international journal. More than neoplastic disease, mostly GI and lung breast. Also, I searched our environment of our hospital. So the HBV is hepatitis, you know hepatitis. So we have a bunch of hepatitis. In Pusan area, they are highly impacted in the hepatitis. I don't know why, but drug abuse in the Pusan area. There are many drug problems. Drug is like cocaine, like marijuana. So they need needles. So because of this needle, the hepatitis incidence is high compared to other areas. So the HBV and compared to 2007 to 2011, the cancer patient is highly increased. So we select chronic liver disease, mostly hepatitis, and the coronary heart disease, and the cancer. Why we select coronary heart disease? Coronary heart disease, do you know? The thrombus in the blood vessel, blocked blood vessel, and heart necrosis, heart muscle die. That is a coronary heart disease. So if the coronary heart disease, we need some stent into the blood vessel, and some instruments, and get the pathway to blood going through. So to get the sample, we need consent from the patient. Consent. It is very important. If we don't have consent, we cannot store or we cannot use biospecimen. But the the cancer and the coronary heart disease, we need some surgery and procedure. Before procedure, we need some consent for the surgery or consent for the procedure. During that time, we just put this consent form for the sample collection. Most people, they don't know what is this. They sign it easily. Can you understand? I can explain in Korean easily, but it's very difficult in English. So that's why we select coronary heart disease and the cancer. So this is at first, this is our strategy. Biobank for the translational research about cancer and the cardiovascular disease and the liver disease. So we distribute the sample to the research, and they do some feedback from the research. We can make improvement, diagnosis and the therapeutics and the new drug and the disease prevention. This is our strategy for the biobank. So this is our action plan. We collect the sample. Mostly, if you think that the biobank is not only sample, but also other information is important. Other information means clinical, disease information, and chemotherapy information, and some epidemiology, demographic data. It is very important. Also, we also collect normal and blood and the disease. So we call it matched sample. So we collect this matched sample, and we distribute to the clinic research for the biomark identification to using this matched sample. They present biomarker to us, and we developed clinical validation and developed the kit. I will show you some example of this next. So we have developed biomarker-based kit. So I briefly our instrument, our people, and our specimen. So this is our structure. Biobank director, not me. I am here. Above me, this. He is a surgeon. Why he is a biobank director? Because after surgery, after take off the specimen from our body, it is very important, golden time. After 30 minutes, RNA degrade. Also, if you want some microRNA, the short RNA study, the vascular clamp to cut tissue, we need a vascular clamp and the cut. When the vascular clamp, the ischemia start. So we need surgeons help so much. So after cut specimen, they send to our biobank immediately. So that's why we promote this surgeon as a biobank. He do nothing in the biobank. I am do everything. But the most important thing is cut specimen and send to us immediately. That's very important for the biospecimen quality. So that's why biobank director is a surgeon. And the research for the QC, and the medical technologist for the correct specimen, and the nurse for the clinical data, and the medical record technician for the clinical record. So we are our team like this. So our biobank is part basement of this cancer center. So near to pathology. Do you know pathology? Really? I am MD and pathologist. Pathology is a kind of a big area. I am GI pathologist. When you go to the hospital, you do some endoscopy and take a biopsy, take a tissue, and the pathologist made some slide. We call it HNE slide, and take a look using a microscope. So we call it is a cancer or not. So if the cancer, to do the surgery like this. So for this, the bank near to pathology. So after surgery, surgeon sent to the pathology for the diagnosis. And after diagnosis, we take tissue from the normal and from the cancer. So we storage in the liquid nitrogen tank like this. And then the QC storage and the distribution. So as mentioned before, the biobank store sample, but the epidemiology data and the clinical data and the demographic data. We also connected to clinical information center in our hospital, EMR. EMR is electronic medical record, so that's an important data system. So the cardiovascular cancer, chronic hepatitis, we collect valuable specimen for the research. So also surgeon is important. They get the consent from the patient. So after selection and the consent, transport specimen, we collect normal blood and the disease and the storage and making TMA. So we have this equipment, storage for the liquid nitrogen tank, and automation and the QC and informatics. So from the 2008, last July, we collect this specimen, 28,000 samples, and the distribution is almost 7,000 specimen. So we distribute hepatitis blood serum to the research. They developed this, and they made this kit for the HPV drug resistant kit. So that's our base success. And for the this is a high impact journal. They use the specimen will provide the National Biobank of Korea in the KAIST. Before this talk, I met this Professor Shin in KAIST. So we distribute him these liver samples for the research. So we can help many researchers for the biobank distribution. So there are online distribution site. You can go to this online one-stop portal. You can easily how many samples how many, which sample is where bank. You can easily see in using this the website. The last is just I present my experience using this cancer genomic research using the Biobank. We have this bioinformatics center, the optometry, ion proton, and the chiasm pyromark for the bioinformatics. So this is not good, but not best, but the good journal. I made some data in here, so published last year. This is the MUC5AC is a gastric machine. So when you drink so much and vomit, how about? The mucus out there, mucus. 끈적끈적. I don't know why it. So the 끈적끈적 is a mucin. The major component gastric mucin is MUC5AC. So this is a various kind of mucin domain. One is secreted mucin, and the other is membrane-associated mucin. So I focused this secreted mucin, MUC5AC. So when we do the immunohistochemistry for the stomach cancer, mostly this MUC5AC expression is a superficial portion of gastric cancer, like this here. But in deep part, if cancer grows deep part, loss of MUC5AC. That means MUC5AC down is increased invasiveness of gastric cancer. It means the same in here. In here, superficial, well preserved MUC5AC in the gastric cancer cell, but in deep portion, like this. So I hypothesized that the MUC5AC decrease is associated with the increase in invasiveness of gastric cancer. How to prove it? So I asked Biobank using the gastric cancer tissue. So this is the clinical data. This is survival data. This low portion is decreased MUC5AC. This upper graph is preserved. So this is the survival rate. So if the decrease of MUC5AC in the clinical data, the more patients die. It means MUC5AC down is poor prognosis in gastric cancer. So we need how MUC5AC related with decreased survival. So we do some cell experiment. What's your topics? Mixed or major? Your major is not only molecular biology, but also other major. So somebody understand and somebody cannot understand, right? So I will briefly explain. So when the MUC5AC down, the increased invasion and the migration of gastric cancer cell, I can see. So also we ask a fresh tissue sample from gastric cancer patient, and we divide between MUC5AC positive and MUC5AC negative. And what's important pathway for this MUC5AC down regulation? We found that this PI3-kinase AKT pathway is important between MUC5AC down and MUC5AC increase. So we do some signal and pathway study like this. When I MUC5AC down, the AKT phosphorylation increase. It means MUC5AC down and the AKT PI3 kinase increase. That's why increase gastric cancer invasion and migration. And finally, patient die. So I'm pathologist. So when I do my practice, so when sign out as gastric cancer, do the immunohistochemistry for the MUC5AC, and when MUC5AC down, I ask a clinician that this patient is bad for the prognosis. You have to more chemotherapy, more extensive therapy we need. So that's Biobank may helpful for the patient management. This is a continuous photograph. And recently, not only cDNA array, but also many NGS technology developed. So next time, I have some plan to do some therapeutic target for the colon cancer. But I have no technology. Also, I have no money for the NGS technology. So I asked some banks, but not the banks. LG Life Technology, someone. So I asked why don't you collaborate with me and some do research. I have a bunch of tissue, you have a bunch of money, and we can collaborate, and we ask somebody to do some NGS, and we can develop some therapeutic targets. So I collaborate like this, LG Life Technology, and for the good result, multi-institution is very important. For the clinical trial, for the new drug, just only one hospital is not enough. For the developed new drug, we need a multi-center study. So two center, Pusan National University Hospital in Gwangju and our hospital collaborate to collect the tissue sample. And for the bioinformatics, Macrogen, the #1 NGS company in the Korea, so and the LG Life Science, we collaborate. I will show you some NGS data, but I'm not the bioinformatician, so I cannot exactly what it is, but I will show you the NGS data. This is a collection of cancer sample and the filtration, and at first we plan to the fusion protein, fusion genes. Why fusion gene? Fusion gene is not present in our normal body. So we made the target for the fusion gene. It is a very important therapeutic target because we have an inhibitor for the fusion gene. It doesn't harm our normal body. So it is very important, the fusion gene. But in colon cancer, there are not many reports about the fusion gene. So we start in fusion gene and the other SNP study and the alternate splicing study. So the whole sample is 180 sample about. So this is RNA-6 throughput. It is enough for the good analysis. So the red dot is normal and the blue dot is cancer. These three samples is outliers. So we delete these three samples and go to the more analysis. is a SNP study, mutation study. Most of the mutation studied by TCGA. TCGA is the American tumor study group. So LG and the TCGA and the GenTech are mostly similar data. So our samples and our method is not bad compared to other study group. So this is a frequency of mutation, TP33 and the APCC, some other mutation. But the mutation is not important. We want to find some fusion genes for the therapeutic targets. So this is a candidate pool for the fusion gene, binding protein, generic enzyme like this. So we found some fusion, LG-LS-001 fusion. This is their patent number. Already we submit and we are revision now. So this is the NTRK gene fusion gene. NTRK is a tyrosine kinase fusion gene. Tyrosine kinase has many inhibitors already in the market, so we can easily develop anti-cancer drug for this fusion gene. So we found this fusion gene, and you can see this fusion in the tumor tissue like this. Although this is very, very complicated, I cannot understand exactly what it means. But this fusion occurred using this interchromosomal deletion or inversion of NTRK genes, like this. This is the architecture of fusion protein. So, also, this is the mutation profile, and this is the fusion gene profile. If some gene mutate, they are oncogenic. But in the fusion gene, the case with the fusion gene, they don't have enough mutation for the oncogenic. So we call it mutually exclusive. So fusion gene itself is meaningful oncogenic without mutation. That's a very important conception. So we think that this fusion gene is fully oncogenic. So in this fusion gene rearrangement reported other cancer like congenital fibrosarcoma and breast cancer, but not colon cancer previously. So we found 2% of colon cancer showed this fusion gene, like this. So, and next step is what is inhibitor? LG Life Science focused on this fusion gene inhibitor. Fusion gene inhibitor is very important. So we searched the Medline and the other resource for this inhibitor, like this, kinase and the product potency and the selectivity, this NTRK gene specific inhibitor. So and now we are focusing on the more potent inhibitor for this fusion gene with the LG Life Science. So this ICISTS-50 is 0.8. So we are focusing on this more potent inhibitor. So you can see the fusion gene by the PC like this. And this is a very important picture for this fusion gene. So I present two sample of using Biobank for the cancer genomic study. we can use this biospecimen using this genomics and the bioinformatics and the functional study and the biomarker and the cancer therapeutics. Also, we can move to the clinical validation and the clinical trial and going now. So the biobank is very important for the cancer